in pH are tolerated better than rapid changes that will cause the cell apoptotic. Jadi kalau rapid changes, of course lah, rapid changes contohnya pada pH 7, dia tiba-tiba turun kepada pH 4 ataupun 3. So ini lebih teruk berbanding dengan kalau dia daripada 7 kepada 6.7, 6.5, 6.3 kan. So the slow changes actually boleh tolerate lagi. Okay, tetapi kalau dia rapid changes, so dia akan lead to the cell apoptotic. And most of the cell will tolerate a medium between the range of 6.5 to 7.8, but medium which is much outside of this range can be lethal, which will lead to your cell death. So the regulations of the pH is done through the variety of the buffering system. Contohnya ada certain fermenter, contohnya kita nak culture kita punya bacterial cell. So some of the fermenter, they are equipped with the buffering system. Okay, they have the alkali and also the acid. So if the pH drop, then the alkali will be added into the medium automatically. Or if the pH mediums rise, then the acid will be added automatically into the media. Okay, so they have the automated system, buffering system, right? But sometimes you don't have this fermenter, but that's why you need to check the pH from time to time to assure that the pH range is within the tolerated pH range by the cell. Okay, so understanding the metabolism of the cell is essential to able to derive the best method to control the pH using the media and also the buffering system available, and also produce lactic acid and so carbon dioxide as a byproduct of the energy metabolism. So generally, as we all know, if the cell is fast growing, then it will produce much more lactic acid and also the carbon dioxide compared to the slow growing or non-growing cell. Okay, so the actual pH in the culture at a given time that's determined by the culture configuration and the media of the cells are grown in the cell types and also the buffering capacity of the medium. So cells are passively controlled by the types of the dish, media composition, the depth, and also the buffer system. So if sometimes we have to be very careful with the depth of the media, because when cell they are the cell which is on the top and the cell at the bottom, they receive different for example gases. Okay, so this also take into account, right? And sometimes the metabolite may sedimented, so they actually will very much affect the cell at the bottom. Right? So cell growth in the fermenter or the instrument spinner have active control pH through the program additions of the acid and base to maintain the pH at a given set point. So we have the spinner. The spinner helps to stir the media so that the nutrients and also pH and also the gas will be distributed evenly to every single cell, right? So high concentration of the bicarbonate of the media formulation require higher carbon dioxide percentage in the air supply to provide an appropriate pH in the medium, but also to provide the greater buffering capacity. Okay, so carbonate ion themselves also they will affect the cell function to some extent that some cells that require bicarbonate to grow independent of its requirement as a buffer. Okay, so the choice of the buffering system will clearly affect both the pH and also the osmolarity of the final medium, as well as other aspects of the cell physiology controlled by the ion's environment. So that's why I keep telling you that you need to understand clearly the characteristic of your cell. As I said just now, some of the cells they prefer lower pH, and some of the cells they prefer higher pH. So understanding the characteristic of the cell will very much helpful to make sure that you culture the cell successfully. All right. Another one is the osmolarity, which is determined by the medium formulations. Okay, and most of the time the salt and also the glucose are the major contributor, and sometimes the amino acid may contribute significantly. Okay, so altering the osmolarity significantly by more than 50 milliosmol a liter will always affect the cell growth and also the function in some manner. So almost all commercial medium are then formulated to have a final osmolarity to around 300 milliosmol. So remember that 300 milliosmol. So significant deviation outside of this range of the optimal will result in the loss of the membrane integrity. So very important to make sure that you have the right osmolarity, and you can check the osmolarity using the osmometer that are available in the lab. And also the adding NaCl, so natrium chloride, can increase the osmolarity of the media, because the natrium chloride will, when they dissolve in water, they will become the iron positive and chloride negative, and this will affect the osmolarity of the medium, as well as the glucose. Okay. And also the oxygen level is generally part of the buffering system of the media and must be set accordingly. So the oxygen level in the incubator are then provided by the 50% of the air in the incubator mix. So the seal box with the entry, the exit point with the premix of the with the oxygen concentration can also be used in the incubator, which is the cheaper way. And of course, some of the fermenter is probably the most accurate way to study the effect of the minor variations of the concentration of the oxygen at the cell function. But too much oxygen also not good, yeah, because they may cause toxic to the cell as well. So researchers are more concerned with the change in the oxygen concentration of the medium generated by the cell themselves and each effect of the cell function. So the common problem is lack of the adequate oxygen in the cell, because most of the cell they need oxygen, okay, and they'll emit the carbon dioxide. So oxygen generally will only diffuse freely through the first few milliliters from the surface. So those reside at the bottom of the media will not receive enough oxygen, and that's why they probably will not replicate as efficient as the cell which is sitting on the top or next of those cells which is reside at the surface. Okay. So among of the mediums in the media, this and the flux will be a balance between the cell needs for the nutrient over the course of the experimental and the rate, which will, oxygen can diffuse from the surface to the cell monolayer. So too much medium will inhibit oxygen diffusions and so and slow cell growth, but too little may be exhausted, leading to the cell death. But that's why you need to be very cautious, yeah. So you don't want too much medium, because too much medium, so those cells reside at the bottom will not receive the oxygen, okay, and it will cause the cell slow cell growth. However, if you put too little of the medium, then the medium may be dry out easily, which the medium nutrients will be exhausted much faster, which will lead to the cell death as well. So you need to plan very well when you culture your cell. Look at the characteristic of the cell, whether the cell is fast growing, non-growing, or slow growing. Okay. And a clear and obvious difference between the in vivo and vitro environment is the surface to which the cell attach, and also the sap that acquire as a result. So the collagens, the laminin, and ferrolactin serve as the component of the extracellular matrix, which is attaching for the cell to grow. This is particularly important for the cell dependent, okay, cell dependent culture, okay, or encourage dependent cell, okay, where they need the culture, need the matrix to attach to. And sometimes serum also contain soluble attachment photos such as the fibronectin, which will rapidly bind to the tissue culture plastic and provide a modified surface for the cell attachment. And most of the cell in animal grow attached to some structure of the connective tissue, basements, the membrane, and also the mineral matrix, such as bone. And cell in the blood, lymph, and other fluids are only those are normally grown in the suspension. Okay, so the shape and size of the cell in vivo, they depend on their environment, what are they attached to, and the hormonal signal and are receiving and the function. So epithelial cell may form a single cell layer attached to the basement membrane with thick and highly polarized cells, whereby the petricular cells may also be attached to the single layer, but they may be very thin, acting more as a physical barrier than the proteins and the mucopolysaccharide secreting factory. So most cells will be rounded when grown in the suspension and flattened when grown in the surface, such as the tissue culture plastic. Okay, do you know why? Are you still with me?
Yes, doctor.
Yeah, doctor.
Why they are rounder when they are grown in the suspension and flattened when they are grown in the culture plastic? Anyone want to try?
In suspension, they have the nutrients all around, but on a flat surface, they only got the nutrients from one side.
Yeah, that's very good. Okay. Or you may look at this analogy. Okay. Class, do you have butt? Ada punggung tak?
Yeah.
Ada kan? Cuba tekan. Punggung tu round ke tak? Round kan? Okay. So when you sit, what happened to the shape of your butt?
Flatten.
Flatten kan? But when you swim in the river and swim in the pool,
Rounded.
oh, see? This is like your shape, the cell shape lah of your butt. Faham tak?
Okay.
Paham.
Senang faham kan? So some cells such as the pancreatic beta cell and the testicular radic cells may show increased function when they are allowed to maintain a rounded shape. Other cell type may need to attach and spread to survive. Okay, again, this is very much depend on the cell character. So plastic or glass surface can be coated with the purified attachment protein, such as fibronectins, laminin, and also collagens to assist the attachment of the cell. And the basement membrane can also be laid down on the culture dish by one cell type, and this cell will remove sterilely, and the second cell type culture on this cell produce extracellular matrix. And a cell can also be selected to produce the tissue type specific extracellular matrix that would be most appropriate for the cell to grow in vitro. That's providing a more physiological relevant culture model. Okay, so the artificial membrane will include the polyethylene tetraphthalate, the polytetrafluoroethylenes, polycarbonate, and also the polyester. These are the very commonly used artificial membrane, okay, in animal tissue culture. Okay, what are the advantages of this artificial membrane? First, it can be used to separate the cell from the media and one cell from another. Secondly, the cell will extend the process, which is the pores of some of the membranes, and even migrate through the membrane if the pores are large enough. And the membrane can be chosen to have a specific molecular weight cutoff, and thus can pass or retain the nutrients or the cell secreted product on the basis of the size. These are the some of the benefits of using the artificial membranes. Stress, okay, as you know, stress also can kill cell directly and set off the natural genetic program that may lead to their death or apoptosis, or cause mutations or other changes that would not reflect the true natural function, functioning in vivo, or their desired function in vitro. So may due to the rapid change, this may due to the rapid change of the pH, the temperature, and also the osmolarity. Now, this is all the physical environment that you have to take into account seriously when you culture yourself. Another one is the mutagenic chemical, or other, even light may also lead to the cell death or mutation. Okay, or digital-like compounds such as twin 80 may also lead to the leakage of the ability of the cell to maintain the pH and also the ion gradients necessary for the function, especially when there are leakage at the membrane bilayer. So all the all the chemicals in the internal compartment will leak out. Okay, this will very much affect the physiological process of the cell itself. And oxidative damage to the cell is also, and it will present threat that the oxygen is also necessary part of the culture environment. So cell may be differentially sensitive to such damage depending on their cytoplasmic enzyme, the cellular composition, and ability to produce protective enzymes such as the superoxidase dismutase. Okay, some of the cells they're able to handle the stress well. Like humans, some of the humans they can handle stress very well, some cannot. Once they come to the small matters, everything, they can stress out. But this is just like cell. Right? Some of the cells they are very strong and they can deal with the stress well. All right. And cell may also sensitive to the contaminants and also breakdown of the products that's found in the medium, such as the heavy metals contamining the medium component. This heavy metal can be found sometimes can be found in the serum. organic compound from insufficiently pure water. Pure water may have some organic compound, or the ammonia produced by a byproduct of the cellular metabolism. And that's why it is important to keep in mind the difference between the goal of recreating an in vitro environment that allows the cell to function well to the greatest extent possible, or the same way that they do in vivo and other equally valid aims of the culture. So when you do cell culturing, always remember that you want to create, or you want the environment that can mimic, okay, the actual environment of the cell in vivo, right? So that the cell will continue to survive and continue to replicate. This is the ultimate goal when we do animal tissue culture, right? So some of the chemical and heavy metal damage cell, if not found in the, if not bound or sequestered, or the serum can also contain toxic substances like heavy metals. Okay, that's why serum load should be tested and then compared using the cell line that you are planning to culture. You must buy the component of high purity and prepare tissue culture grade water in the laboratory for the making media, and keep the glassware and weighing devices for use exclusively in making media. So always remember, you don't mix all these devices. Okay, if you mix them, then the chances to get contaminations is very high. Okay, and protease also can also be secreted by a viable cell or released by a dying cell into the media, because this cell can directly injure the cell, or they can remove the cell surface components such as the receptors that are necessary for the proper cell growth, or they may degrade the growth factors adding to the medium or the protein secreted by the cell. Okay, so protease also one of the factors that will lead to your failure of your culture. And the summary of this unit is that the best control physical environment still needs the additions of the well thought out chemical and also hormonal environment to produce the optimal growth and the functions of the cell in vitro, right? And I think this is a slide, last slide. And I think that's all for today. Do you have any question?
Doctor,
Yeah.
I don't understand how high oxygen can be toxic to the culture.
Why not? Everything which is excessively extra will cause bad effect to the cell. Like glucose. If glucose too high, glucose is a food for the. Okay. May I know who is on the line, please?
Oh, Adlin.
Adlin. Okay. We need glucose kan to generate kita punya energy kan.
Yeah.
Yeah. But how about if too high glucose?
Fat.
Not only that, it will cause diabetes, right?
Yeah.
Yeah. Same with the oxygen. Because kita when we culture the cell kita ada dua tank, which is the oxygen tank and carbon dioxide tank, which will provide the gaseous surface to the cell. But sometimes when you provide too much oxygen, so it will cause toxic to the cell because it's too much oxygen. They need oxygen, but too much, they will cause toxic. Like our body also, we need glucose, but too much glucose, it will cause diabetes. Faham tak?
Faham.
Sama lah. We cannot, let me tell you, all the organism system they need moderate monitoring, and then they only need moderate punya supply. Just enough is good enough. Tapi tak boleh excessive. When they become excessive, then something will go wrong with the system. This is the rules of the organism.
Okay, thank you, Doctor.
Alright. Ada lagi soalan? Kalau tak, then we call it a day, boleh? Siapa nak scan? Siapa nak scan attendant? Ada tak orang nak scan attendant?
Saya, Doktor.